Hello everyone, I am Arpan, PhD student in Faculty of Biology, Jagiellonian University in Krakow, Poland. The topic of my presentation is robust estimation of cellular features and data analysis in confocal micrographs from Arabidopsis thaliana seedlings. In microscopy, the form of data is images, and the way of representation is visual comparison, and sometimes fluorescence intensity is used to compare between the designated channels. However, the underlying morphological features of the image pixels are often underestimated. Morphological features can be determined by estimating the differences within the pixels. In plant sciences, morphological properties of the cellular and subcellular compartments are important from structural and functional point of view. In order to find the morphological diversity, we propose a pipeline where we estimated the morphological parameters like heraldic features, spatial features, and intensity features from the micrographs. We estimated these features in wild type, the null mutant, and the mutants that are reported to have irregularly shaped cellular compartments called ER bodies in Arabidopsis thaliana seedlings. It has been observed that in plants of Brassicaceae family, there are ER-derived cellular compartments called ER bodies. In transgenic lines of GFPH, these emit most of the GFP signals, revealing the shape and the complexity of the structure. Coming to the constituents of the ER bodies, Ni1 is the major transcription factor that regulates the formation of the components that assemble together to form the ER bodies. Ni2 being the scaffolds protein, pipetane is an homologue of beta-glucosidase enzyme, with MEB1 and MEB2 proteins that are integrated into the membrane. ER bodies activate plant's defense system, popularly known as mustard oil bomb system, by hydrolyzing the substrate glucosinolase. In order to set up the experiments, we obtained 7-day-old seedlings from plants of wild type and mutants. We used propydium iodide staining to distinguish the cell wall from the ER bodies, and use constant settings for taking the micrographs. In our micrographs, we take a series of images representing the 3D view of the objective. We use max contrast projection to merge these slices of images together, and these merged images were used to segment the cells from each micrograph. Features were extracted from the segmented cells individually, and parameters that defines the spatial intensity and heraldic features were estimated. From these, we obtained a feature matrix with sample IDs corresponding to micrographs and segmented cell IDs from each micrograph and their corresponding features. The feature matrix holds the parameters that defines the morphological features. These metrics can be used further to conduct sample-wise analysis, segmented cell analysis, and relationship between the cells and their organelles. Coming to the results from 2 experiments that we performed, we used Pearson correlation distances on the normalized feature matrix, both at sample and segmented cell level. Our MDS analysis reveals that the morphological variation in ER network and ER bodies explain maximum variation in the MDS1. And in MDS2, the maximum variation is explained by the lack of function in the mutant. One thing to note is the mutant 1 and mutant 2 are different. Mutant 1 has ER bodies but morphologically distinct cells, and most of the samples in mutant 2 were reported to have larger cells but no ER bodies. Our morphological analysis therefore holds true for ER body mutants. We used normalized feature matrix to compute k-means clustering and obtained groups of segmented cells that are similar to wild type and ER network. We considered that the cluster representatives of the cells that are dissimilar to the null mutant, and further looked into the cluster representatives and observed that mutants indeed show morphologically distinct ER bodies compared to that of wild type. By discriminant analysis, we found that the ER body mutants have proportion of cells predicted to be similar as observed genotype, about 70%. The feature matrix revealed that the parameters have a distinct profile than that of wild type. The sample-wise and the segmented cell-wise analysis shows consistency in the estimation of these parameters and their relationship. Comparative statistics using multiple linear regression shows all genotypes have distinct morphological properties compared to that of null mutant, and within their body mutants there are significant differences in the parameters observed at FDR less than 0.01 when compared to wild type. All in all, the morphological variations are difficult to estimate, but our pipeline sort of estimates the special heraldic and intensity parameters and determines the morphological diversity. And we could distinguish between the ER bodies and ER network quite nicely. And finally, we have observed the MEB1 and MEB2, which are involved in the membranes of the ER bodies, has some importance on the morphological properties of the ER bodies. So with this, I conclude my presentation. Thank you for your attention, and please